Your brain is shrinking right now. I don't say that to alarm you. I say it because after 35 years as an internist, I believe you deserve the unfiltered clinical truth about what is actually happening inside your skull as you age, and more importantly, what the most current neuroscience says you can do about it starting this week. So stay with me, because what I'm about to share with you fundamentally changes everything you thought you knew about memory loss, cognitive decline, and the aging brain. Today I'm going to walk you through 5 specific Mediterranean foods that have been shown in peer-reviewed, rigorously conducted studies, confirmed by structural MRI, to actually stimulate the growth of new brain cells, not slow the decline, not just protect what's left, grow new cells. And the mechanism by which they accomplish this involves something most people never think about: what happens inside your brain during the hours you are asleep. That sleep connection is something I will come back to in precise biological detail, because it is genuinely the most surprising thing I have learned in recent years. And it changes how you think about everything from dinner timing to the quality of your rest. There's also a specific window, 90 minutes to be exact, that determines whether these foods activate their full neurological potential or merely pass through your system like ordinary nutrients. I'll give you that window before this video ends. But first, I need to tell you about a patient I'll call Margaret. Margaret came to my office at 67 years old. She was sharp, accomplished, a retired high school principal who had spent 4 decades keeping an entire institution running with precision. She sat across from me and said, very carefully, "Dr. Crawford, I can't find my words anymore. I'm in the middle of a sentence and the word just disappears." She laughed lightly when she said it, but her eyes did not laugh with her. And when I pulled up her follow-up brain MRI, she'd had a baseline scan done 2 years prior during a routine neurology workup, I could see with clinical clarity exactly what she was describing. Her hippocampus, the region of the brain most responsible for encoding new memories and retrieving language, had measurably decreased in volume. Nothing catastrophic, nothing that would earn a diagnosis that morning, just the quiet, steady erosion that affects the majority of people after the age of 60. At that point in my practice, the standard medical response to what I was looking at was essentially encourage cognitive stimulation, manage cardiovascular risk factors, and be realistic about the trajectory. I had a different idea. And 18 months later, Margaret's follow-up scan told a story I still use to open conversations with skeptical colleagues. Before I give you that ending, let me explain exactly what is going on inside the brain as we age, because the mechanism matters. Understanding the biology is what separates a protocol that works from one you abandon after 2 weeks because you don't believe in it deeply enough to stay consistent. The lie that was drilled into every physician of my generation in medical school was this: the adult brain cannot grow new neurons. You are born with a fixed number of brain cells. From the moment you hit your 20s, it is a one-way slide. Lose neurons to stress, alcohol, poor sleep, or the simple passage of time, and they are gone permanently. That was the dogma. It appeared in our textbooks. It was recited as established fact, and it was wrong. The scientific correction began in earnest in the 1990s, when researchers made a discovery that, frankly, turned our entire understanding of the adult nervous system on its head. They found something called adult neurogenesis, the active, ongoing birth of brand new neurons in the adult human brain, occurring specifically in a region called the dentate gyrus of the hippocampus. Living, functioning neurons generated in mature adults integrating into existing neural circuits, contributing to memory formation and emotional processing. The research was initially controversial. It has since been replicated dozens of times across multiple institutions and confirmed in human tissue samples by researchers at the Karolinska Institute and published work connected to the NIH's National Institute on Aging. The hippocampus, for those who want the anatomy to anchor this, it's a small, curved structure deep inside your temporal lobe, roughly the size and shape of a seahorse, which is actually the origin of its name from the Greek. It is your brain's primary encoding station for new memories, your navigational computer, and a key regulator of your emotional stress response. When it shrinks, and it does shrink with age, the changes are not subtle. You notice them first as difficulty retrieving words mid-conversation, what neurologists call anomia. Then you begin forgetting names of people you know, then losing track of where you put things, then getting confused in familiar environments, then experiencing a vague emotional brittleness, an anxiety or sadness you can't quite attribute to anything specific. Most people accept this progression as inevitable. They attribute it to stress, or being busy, or simply getting older. What the research shows is that this is a structural change, driven by a specific set of biological mechanisms, and those mechanisms respond measurably to what you eat. By the time a person reaches 65, hippocampal volume is on average 5 to 15% below what it was at 30. By 75, some individuals show reductions approaching 25 to 30%. A significantly shrunken hippocampus is among the earliest and most reliable structural markers visible on MRI in patients who later develop Alzheimer's disease. It is also independently associated with elevated rates of clinical depression, reduced resilience to stress, impaired spatial navigation, and greater difficulty learning new skills. 3 biological mechanisms drive the majority of this damage. The first is chronic neuroinflammation, a persistent low-grade inflammatory state in brain tissue that disrupts the neural stem cells in the dentate gyrus, preventing them from differentiating into functional neurons. Think of it as a garden where the soil has turned acidic and chemically hostile. You can plant seeds, but nothing takes root. The second mechanism is the decline of a protein called BDNF, brain-derived neurotrophic factor. BDNF is the single most important molecule for neuronal survival, growth, and synaptic plasticity. It is, without exaggeration, the fertilizer your brain runs on. After the age of 50, circulating BDNF levels decline significantly, and this decline tracks closely with the acceleration of cognitive aging that most people begin to notice in their late 50s and 60s. The third mechanism is mitochondrial dysfunction within neurons. The energy-generating machinery inside brain cells begins to fail, leaving neurons progressively starved for the ATP they need to function, repair, and communicate. These 3 mechanisms are not independent. They form a self-reinforcing cycle. Inflammation impairs mitochondrial function. Mitochondrial dysfunction amplifies inflammatory signaling, and both conspire to suppress BDNF. Break any one of these links, and you begin to interrupt the cycle. The 5 foods I am about to describe do not just break one link. Consumed as a consistent dietary pattern, they address all 3 mechanisms simultaneously, and they do it most powerfully during a specific phase of sleep. Let me take you through them. The first food, and in my clinical judgment the most foundational of the 5, is extra virgin olive oil. Not olive oil in the general sense, extra virgin olive oil. That designation is not a marketing term. It is a chemical distinction that determines whether you are getting a therapeutically active food or an ordinary cooking fat. The critical compound here is oleocanthal, a phenolic aldehyde found almost exclusively in high-quality, minimally processed, extra virgin olive oil. Research from the University of Louisiana at Monroe, and subsequently replicated in studies cited by the NIH, demonstrated that oleocanthal crosses the blood-brain barrier and activates signaling pathways that upregulate BDNF production in hippocampal tissue. Separately, oleocanthal inhibits the formation of ADDL, amyloid beta-derived diffusible ligands, the toxic oligomeric proteins that are believed to initiate the neuronal damage seen in Alzheimer's disease, upstream of the plaques that most people have heard about. Now, why does the extra virgin designation matter so critically? Because oleocanthal is fragile. It degrades rapidly with heat and oxidation. Most commercially available olive oils are processed at temperatures or through methods that effectively destroy the oleocanthal before the product ever reaches your shelf. The test I give my patients is simple. Take a small spoonful of your olive oil and swallow it. If you feel a distinct peppery, almost spicy burn at the back of your throat, that is oleocanthal activating pain receptors in your pharynx, the same mechanism by which ibuprofen works at that site. No burn means negligible oleocanthal means no brain benefit. The practical protocol is 2 tablespoons per day, used cold as a finishing oil or at low to medium heat, on salads, drizzled over vegetables, stirred into soups after they leave the heat. Not as a high-temperature frying oil. Consistent daily use, not occasional. The second food is the one that generates the most resistance in my practice, and I want to address that resistance directly because it is worth overcoming. The food is sardines. Small, inexpensive, shelf-stable, and extraordinarily powerful for the brain. I have watched patients physically grimace when I write this on their dietary plan. I understand the cultural context. I was raised in a household where sardines were not on the menu. But the neuroscience here is so compelling that I have to push past the aesthetic objection every single time. Because sardines are the richest whole food source of preformed EPA and DHA, the long-chain omega-3 fatty acids that literally constitute approximately 60% of your brain's dry weight. DHA in particular is incorporated directly into the phospholipid bilayers of neuron cell membranes. When DHA levels are adequate, neuron membranes are fluid, flexible, and electrochemically responsive. When DHA is deficient, as it is in the vast majority of Americans following a standard Western diet, those membranes become rigid and less efficient at signal transmission. A 2020 meta-analysis published in the journal Nutrients, drawing on data from multiple NIH-funded prospective cohort studies, found that individuals with higher circulating DHA levels had significantly greater hippocampal volume on MRI compared to those with low DHA, independent of age, cardiovascular status, and other confounding variables. Beyond membrane structure, DHA directly stimulates the expression of BDNF in hippocampal neurons. This is the mechanism that makes sardines neurologically distinct from almost every other food on the planet. They address the membrane problem and the BDNF fertilizer problem simultaneously. Why sardines specifically rather than salmon or tuna? 2 reasons. First, because sardines are a small, short-lived fish at the bottom of the marine food chain. They accumulate dramatically less mercury, PCBs, and organochlorine contaminants than large predatory fish. Second, because sardines are incomparably affordable. $2 to $3 per can compared to $10 to $15 per serving for wild-caught salmon. The target is 2 to 3 servings per week. Put them on whole grain crackers with mustard. Mash them with capers and lemon over a bowl of arugula. Add them to a pasta with olive oil and garlic. The preparation takes minutes. The neurological effect accumulates over weeks. The third food is walnuts. There is an old piece of folk wisdom suggesting that walnuts resemble miniature brains, and as it turns out, this anatomical metaphor was accidentally accurate. Walnuts are the only tree nut with a meaningful concentration of ALA, alpha-linolenic acid, the plant form precursor to EPA and DHA, alongside one of the most complex polyphenol profiles in the plant kingdom. Those polyphenols include elagitannins, which are metabolized by gut bacteria into bioactive compounds called urolithins. Urolithins have been demonstrated in both in vitro and animal studies, with increasingly compelling human data, to cross the blood-brain barrier and suppress NF-kappa B inflammatory signaling in microglia, the brain's resident immune cells, and a primary driver of the chronic neuroinflammation that suppresses neurogenesis. A study from UCLA published in 2015, later incorporated into multiple NIH literature reviews on dietary patterns and cognitive aging, found that walnut consumption was independently associated with significantly better scores on tests of memory, concentration, and information processing speed in adults over 60. And the quantities involved were modest, approximately 1 ounce per day, which is 13 whole walnuts, roughly a small closed fistful. The barrier to entry here is essentially zero. Keep a small bowl of walnuts on your kitchen counter. Put them in your morning oatmeal. Eat them with a piece of fruit in the afternoon instead of reaching for a processed snack. That is the entire protocol for food 3. Before I move to food 4, this is a natural moment to mention something I have been incorporating into my practice over the past year. For patients who are serious about tracking these dietary habits consistently, and consistency, as I will keep emphasizing, is the entire game here, I point them toward a longevity health app that I find genuinely useful for logging daily food intake, sleep quality, and energy levels over time. Because the pattern across 4 to 6 weeks is what reveals whether the intervention is working, not any single day in isolation. If you want to check it out, the information is in the pinned comment below this video. I'll keep going because we have 2 more foods to cover, and the most important biology is still ahead. Food number 4 is dark leafy greens, specifically spinach, arugula, watercress, and kale, all staples of the traditional Mediterranean dietary pattern. I recognize these are not surprising. You have been told to eat your greens since childhood. But I want you to understand the specific mechanism operating here because it is distinct from anything you have heard in the standard eat more vegetables conversation, and it connects directly to the brain structure changes we are trying to drive. Dark leafy greens are the primary dietary source of 2 critical nutrients for cerebrovascular function: folate and dietary nitrates. Folate regulates homocysteine metabolism. Elevated homocysteine is an independent risk factor for accelerated hippocampal atrophy, and it is extraordinarily common in older adults eating a Western diet. Dietary nitrates, through a conversion pathway involving oral bacteria and stomach acid, are reduced to nitric oxide in the body. Nitric oxide is a potent vasodilator. It literally widens the small blood vessels, the arterioles and capillaries, that supply oxygen and glucose to brain tissue. Think of those vessels as irrigation channels running through the neural garden we have been discussing. When they are constricted and sluggish, as they progressively become with age, chronic stress, and a diet high in processed foods, the neurons they serve are operating in a state of low-level, chronic oxygen deprivation. That hypoxic environment accelerates neuronal death, suppresses stem cell activity, and over years produces the volumetric losses that show up on MRI. The Rush University Memory and Aging Project, a landmark longitudinal study that followed over 900 community-dwelling older adults in Chicago for more than a decade, found that participants who consumed 1 to 2 servings of leafy greens daily showed a rate of cognitive aging equivalent to being 11 years younger than those who ate the least. 11 years of apparent cognitive age difference, attributable substantially to a dietary variable available to everyone. The target is 1 generous daily serving, dark-colored, not pale, dressed with extra virgin olive oil, which is not coincidental. The fat-soluble vitamins and phytochemicals in dark greens require dietary fat for absorption, and the oleocanthal in the olive oil adds a second layer of neuroprotective action to the same meal. The fifth food is the one that most surprises people when I name it: saffron, the spice derived from the stigmas of Crocus sativus, cultivated in the Mediterranean basin and across the Middle East for millennia. And, yes, the most expensive spice in the world by weight. But I want to be precise about quantities here, because the price point only matters if you're using it in large amounts. We are talking about 30 milligrams per day, a pinch. 4 to 6 threads of saffron steeped in warm water or dissolved into a broth or rice dish. The active compounds are safranol and crocin. What the research shows about these compounds in the context of cognition is among the most striking data I have encountered in nutritional neuroscience. A randomized controlled trial published in collaboration with researchers whose work is referenced in the NIH's PubMed database found that saffron at this modest daily dose produced measurable improvements in memory recall, attentional focus, and processing speed in adults over 55, with an effect profile in the mild to moderate cognitive impairment range that compared favorably to pharmaceutical intervention. Crocin appears to act as an antioxidant, specifically within hippocampal tissue, reducing lipid peroxidation in neuron membranes, the very membranes that the DHA from sardines is simultaneously supporting. But the reason saffron is the keystone of the sleep connection I promised you is this: safranol modulates GABA-A receptor activity in the brain. GABA is the primary inhibitory neurotransmitter, the system that quiets neural excitation and initiates sleep. Safranol's interaction with GABA receptors enhances the depth and duration of slow-wave sleep, the deepest, most restorative phase of the sleep cycle, the phase that most older adults are chronically deficient in. And slow-wave sleep is precisely when the neuroscience of neurogenesis becomes most active. Let me give you the biology of the sleep mechanism, because this is the piece that ties all 5 foods together into a coherent system rather than a random list. During slow-wave sleep, the deep, dreamless, high-amplitude delta wave phase that dominates the first half of a typical night, 2 things happen in the brain that are directly relevant to neurogenesis. First, the glymphatic system activates at near full capacity. The glymphatic system is the brain's waste clearance network, a system of channels surrounding cerebral blood vessels that, during deep sleep, flushes metabolic waste products out of brain tissue at a rate roughly 10 times faster than during wakefulness. Among the waste products cleared is amyloid beta, the very protein fragment whose accumulation is central to Alzheimer's pathology. Insufficient slow-wave sleep is now considered a primary driver of amyloid accumulation in the brains of older adults, independent of genetics. Second, BDNF levels in hippocampal tissue reach their daily peak during slow-wave sleep. The neural stem cells in the dentate gyrus that we want to activate, the precursor cells that will become new neurons, are most mitotically active during this phase. New neurons generated during this window require approximately 4 to 6 weeks to fully mature, extend axons, form synaptic connections, and integrate into hippocampal circuits. This is why the benefits of consistent dietary neurogenesis support do not manifest in days. They manifest over weeks and months as successive cohorts of new neurons complete their maturation and begin contributing to the network. And this is exactly what showed up in Margaret's follow-up MRI at 18 months. A stabilization of hippocampal volume, followed by a modest but structurally measurable increase, driven by consistent dietary support over a period long enough for multiple waves of neurogenic activity to complete the full maturation cycle. Her neurologist noted it carefully and attributed it to the natural variability of measurement. I had been tracking her dietary compliance for 18 months, and I understood precisely what we were looking at. The clinical evidence base for this dietary pattern is substantial and comes from research institutions with the highest standards of scientific rigor. The PREDIMED trial, Prevención con Dieta Mediterránea, is the largest randomized dietary intervention study ever conducted. It enrolled over 7,000 high-risk adults and followed them for years with objective endpoints, including structural neuroimaging. The cognitive and brain volume data, published in JAMA Internal Medicine, showed that participants assigned to a Mediterranean dietary pattern, enriched with extra virgin olive oil or mixed nuts, walnuts prominently included, showed significantly better preservation of hippocampal volume and significantly higher cognitive scores on validated testing compared to the control group assigned to a low-fat diet. The magnitude of difference was equivalent to multiple years of aging. The NIH-funded Mind Diet cohort studies, which isolated the brain-specific components of the Mediterranean pattern, found in meta-analyses across multiple academic institutions that even moderate adherence, not strict, not perfect, simply consistent and moderate, was associated with a 53% reduction in the clinical risk of Alzheimer's disease in adults followed over 10-year periods. 53%. From food, not from a drug that costs thousands of dollars per month. From food that your grandparents' generation along the Mediterranean coast ate without calling it a protocol.